This is the first part of a set of interconnected lectures where we're going to start really large and then we're going to focus down. The first set of this, we'll be talking about genomic complexity. Then we will move after that into actually focusing down on what is a gene. Uh, so genome complexity, I've sort of alluded to this before when I talked about chromosomes and several times, and I said, on a chromosome itself, the DNA that's there, at least from the terms of the genes that are there, are not just all grouped in a nice little orderly fashion. It's not much like you have 1, 2, 3, 4, 5. Now, in some places you do, but in reality, there are gaps and open spaces and all sorts of things. And I alluded to the fact when you're flying over some areas, in a small plane, you see cities and areas that are dense, and then there'll be areas where there's less. Well, it's the same way as far as relatively how genes are packed on chromosomes. Now, as far as genome complexity, we really are moving into the world of modern art here. I mean, you're going to find that, and we're learning more and more about this, how the pieces fit together is really a fairly complex system. It's our old naive view that it literally was gene, kind of a little bit of stuff, a gene, some control region, another gene, is basically out the window. Okay. But to get there, I think we need to do what we've done many times and kind of talk a little bit about the history of how we understood this and the questions that people started to ask. So we're going to spend just a moment at doing that, right? So this is our ideogram, right? We've seen these before, and I mentioned before that on these ideograms, besides the ordering them based on centromere location and overall length, that they're also ordered based on these bands that you see. These light bands and these dark bands tell you that this chromosome belongs with this homolog versus with this one over here because they have a different banding pattern. This is known as chromosome banding, and it's a technique that was developed in the 1960s. And what it does is it allows you to just basically do a fairly simple stain, usually a Giemsa stain or some other stain, that's where the DNA actually takes up that stain. So in areas where you have dark areas here, okay, those tell you about the degree of condensation that we have. And it turns out that as we pack up these chromosomes, we go through G2 and we pack them all down, they don't just pack perfectly square and equal. And anybody that's tried to stick a sleeping bag and a stuffed bag knows that you're going to have some areas where they're really poked down and the other areas where they're a little less compressed. Or if you're dragging all your laundry out to wash it in a pillowcase, you know that at first you put it in as pretty dense and then you really have to push it in, and that's kind of what happens. We have dense areas and light areas. Okay, so the light areas tell us that we've got a little less activity going on there as far as compaction. The dark areas are areas where you've got more uptake of these stains, and therefore you've got more compaction. So in a big sense, just in a broad sense, when you started doing this experiments and they started looking at them, they noticed that they're really kind of two areas, right? They're light staining areas and dark staining areas that you have on, and some chromosomes have a lot of light and not much dark. Some have a lot of dark and only a little bit of light, okay? And it just varies across chromosomes, and it varies across species and all sorts of things, okay? So just sort of on this crude recognition that there were light and dark areas, they divided chromatin into two primary types. The first type is called heterochromatin, and that's going to be these dark staining areas, areas that take up a lot of stain. And then there's going to be euchromatin, that'll be these lighter areas that we have. Okay? So what do we know about heterochromatin? Well, as I said, it's tightly coiled, and certainly during these late prophase, these are metaphase chromosomes, right? So therefore, it takes up a lot of stain, and it becomes dark staining. For the most part, it's non-coding. Now we have to be very careful about that because, as we'll see in a moment, there are some specific areas of heterochromatin which are definitely non-coding, meaning it doesn't have a lot of active genes in it, but it can go in and out of that phase. So while it's non-coding, while it's heterochromatin, it can expand out of that and become coding. But for the most part, this would be non-coding in the sense that there are relatively few genes there. Specifically, when we talk about kind of chromosomal structures that persist for long term, centromeres, these dense areas right here and right here, are areas that remain fairly tightly coiled even in G1, and there are not a lot of genes located right in the centromere areas, nor in the telomere areas, these darker areas on the outside where the chromosomes are protected. There are not a lot of genes found in those areas. You have to come in from the telomeres to actually find it. And those stay pretty much as heterochromatin. Some of these other areas will actually go in and out of heterochromatin. But for the most part, what we're talking about here was areas that pretty much stay densely packed throughout the cell cycle, at least a 30 nanometer fragment. So we're talking about at least solenoids or some of those other forms. So we've got a collection of nucleosomes together, and they pretty much stay that way. Okay. Now, the heterochromatin, because it either is fairly constantly that way or can go in and out, has two names. One is constitutive heterochromatin. Excuse me. These are areas that are always, constitutive means always. So these are areas that always remain fairly tightly coiled, no matter what part of the cycle we're in. Certainly centromeres and telomeres are good examples of these, and there are not regions where you're going to have any coding. In other words, you're not going to have genes that are going to produce protein or message found here. They tend to be areas of structural function to help the chromosome in its maintain its shape or parts. Or they have other activities such as centromeres. Obviously, during mitosis or meiosis, these are the areas where you get all these spindle fiber connections and all those important things going on. Okay. Now they're basically in that form, and then we have what are ones that can move in and out. These are known as facultative heterochromatin, and some examples are like the bar body. The whole bar body is a classic example of an entire chromosome that's become non-coding. Yet, if you need that, actually can expand it out and use it. So these are considered at least one level of being facultative heterochromatin. But it turns out that you don't have to be an entire chromosome to be facultative. There are lots of areas within every chromosome that go in and out. There's some new ideas, and the ideas have been around for certainly over a decade, but new in the sense of trying to apply it to mammalian systems. But based on work that was done in yeast and certainly in plants, there is this idea of RNA-induced transcriptional silencing, which means there is very tiny pieces of RNA that are produced that never leave the nucleus, and what their job is to actually go in and deactivate some and regions of the DNA so that they become densely coiled and packed up. And then when they're needed, they can free those areas up. So there's thought that potentially these are one of the ways. It certainly seems to be the way in yeast and plant, at least in some plant genome parts, that facultative works. They're it's sort of rolling condition. They're pretty much in heterochromatin, but when they're needed, they pop out really quickly, and then they go back into heterochromatin. And probably it's some RNA type transcriptional elements that help. It's questionable whether it actually occurs in mammals that way at this point, to my knowledge, but it seems likely that given the broad spread across these eukaryotic systems, that it most likely does occur in mammals and other systems as well. So it's maybe one mechanism by which you can go in and out of this facultative state. Well, for us, euchromatin becomes a more interesting one because this is where our loci, if you will, and these genes that we've been talking about, these tend to be the less densely packed areas. So we would say these are areas of euchromatin here and euchromatin down here, and then certainly an area of euchromatin here and there, etc. Okay. This tends to be definitely where coding sequences are. In other words, DNA that's actually a gene or a locus tend to be found more in these areas than they would be, say, in these areas. Again, though, you can go in and out of these phases. Okay? These areas tend to be tightly coiled only during late prophase, early metaphase. And these would be the areas most certainly that would become beads on a string type things. Once we get back in G1 and we open our DNA up, our chromosomes up, then these are going to definitely be those areas that are less compacted. Okay, so just a quick summary. This was a short part just to introduce it, and then we'll come back and we'll talk more about how we understand this to work and how our genome is packed in the next little lecture. But for now, we're just a quick summary of what's happening. First, chromatin exists in more than one form, right? In at least two forms, and probably many other variants of that. Heterochromatin and euchromatin. Now it's important to understand that these are continua. Sort of like when we talked about linkage, we talked about we were you were completely linked, or you were independently assorting, and then there was everything in between. We call it incomplete. Well, same thing here, heterochromatin and euchromatin. There's some stuff that's strictly heterochromatin, some stuff that's strictly euchromatin, and then there's things that kind of wave in the middle depending on what cell line you're in, what stage of development you're in, et cetera. They can move back and forth. So it's part of a continuum. Right? And then how do we know this, at least that they exist? Because we have these staining patterns that have been around now for about 50, 60 years, and we can look at this underlying structure and coiling. So they tell us about what's going on because of the amount of dye that they would uptake. Right? All right, well, those two very important terms and ideas. Now let's take that and move it to, okay, we've got some of that. What is all this euchromatin and all this heterochromatin? How do we is it all the same in every plant and animal, or is it, in fact, what can we say about it? That's going to be in the next lecture.